Okay, good morning, good afternoon, everybody. Welcome to this session where we will be talking a lot about fossils, patterns, and how to understand and use all this wealth of information that past biodiversity can actually tell us. So it's my pleasure today to introduce you Moriaki. He is an associate professor at the School of Biological Science and the Swire Institute of Marine Science at the University of Hong Kong. He has a really broad interest in paleoecology and macroecology, especially using very well dated and known macropaleontological records. He will be talking as well about his recent research that has focused on the spatial temporal dynamics of large-scale biodiversity patterns and the climatic and temperature impacts on species diversity. He also has a lot of interest in applying this microfossil data and knowledge to conservation questions. So in this talk, Moriaki will overview the development and recent advances in this time machine biology as a synthetic science of ecology, paleoecology, and biogeography. So I really hope that you will enjoy this talk. And we are planning to organize this session in this way. So the first 40-45 minutes, Moriaki will be giving the talk called Time Machine Biology, Fossils and Biogeography. And then hopefully we'll have 15-20 minutes time period to discuss and ask lots of questions about all this information that he will give us. There are different ways of participating in these questions, and we will repeat this information when the talk is finished. But basically, there are two options. You can raise your hand and talk, or basically, you can use a question and answer icon that you see on the bottom of your Zoom software. So I leave you now with Moriaki Yasuhara, that he will start sharing the screen. Thank you so much. I'm very happy to see all of you here.
Yeah, thank you very much, Sandra. Great introduction. So let's start. Should be this one.
Yes, all looks okay.
Okay, good. Yeah, thank you very much for great introduction, Sandra, and very welcome to my talk about time machine biology. Thank you for your joining us. And I'm talking basically about paleontology to reconstruct past biodiversity pattern with time and space, especially using ocean drilling samples and microfossils like as you see here. And let's start from broad picture about the entire Cenozoic things, what we know. And now, because of the development of ocean drilling, we know quite well about tectonics, climatic, and biotic change or event. And this is summary figure showing such thing, entire Cenozoic from 66 million years ago until present day. And this is famous Jacob's curve showing climatic change. So this side is warmer climate, and this side is colder climate. So Cenozoic time is basically gradual cooling with time, with some exceptionally warmer time. And major climatic event we know, including rapid warming here, so-called Paleocene-Eocene thermal maximum, PTM. And Eocene was super greenhouse time, pretty warm, especially during early Eocene climatic optimum. And after that, we have sudden cooling here during Eocene-Oligocene transition, EOT. And after that, we have a bit warmer time again during Miocene, including mid-Miocene climatic optimum. And after that, we have gradual cooling continuously, And this is close up over here, the last 5 million years. And we, after relatively warm Pliocene, we have rapid cooling again and stronger glacial interglacial cyclicity, especially after mid-Pliocene transition and mid-Brillion event. We have really large-scale glacial interglacial cyclicity. And we know also plate tectonics and continental arrangement at that time. For example, during around Eocene, Oligocene time, both Drake Passage and Tasman Passage open, somewhere like here. So Drake Passage open here, and Tasman Passage open here. So we have sort of Antarctic seaway, and it's an initiation of present-day time basic deep sea or ocean scheme. And after that, gradually Australia is going north, and until Oligocene, Miocene time, Australia reached to the almost present time position and creating massive shallow marine area between northern Australia and southeastern Asia. It's a famous biodiversity hotspot, Coral Triangle. And also, India is gradually going north with time and eventually collide with Eurasian continent somewhere around Oligocene time. And more recently, we have final closure of Central American seaway, even shallow marine connection closed at around 3 million years ago. And associated with this kind of tectonic and climatic events, we have biotic extinction and faunal turnover and changing in biodiversity hotspot. Like we have tropical biodiversity hotspot in Western Tethys during Eocene, so present day Mediterranean region around here. But during the closure of Tethyan Seaway, hotspot is shifting to Arabian Peninsula, and eventually in the Australian archipelago is a Coral Triangle hotspot present day. And during critical climatic events, we have big extinction of deep sea benthic foraminifera during Paleocene-Eocene boundary, and also big faunal turnover during Eocene-Oligocene cooling. And more recently, after closure of Central American Seaway, a bit later, we have big extinction in Caribbean Sea. We are not so sure about the reason why such a delay after the closure. Anyway, also we have climatic transition of mid-Pliocene transition and mid-Brune event. We have strong turnover or extinction of foraminiferan and ostracods. So we know this kind of general big picture of a Cenozoic biotic and climatic events. And it's mostly from our knowledge based on sediment core and ocean drilling. It's a field rapidly developed in past 20, 30 years. And sediment core is like this beautiful cartoon showing what is sediment core and the resulting analysis. So basically we get cylindrical section of ocean floor, and ocean floor surface is present, and deeper sediment is deeper past. So we got core like this, and we cut into the sections and analyzing from older one to new one, reconstructing past change in climate or ecosystem continuously. And this is one example of deep sea sediment core from Sea of Japan showing beautiful black and white alternation. It's basically showing oxygen content change. And this kind of deep sea drilling have quite nice global coverage. Like this is one example of international drilling program. It was originally DSDP and name changed into ODP, and now it's IODP, International Ocean Discovery Program. And they have hundreds or even more than thousands of sediment core from all over the world ocean, including Indian, Pacific, Atlantic, and Southern Ocean, even Arctic Ocean, we have some good bunch of sediment core. So we have kind of global coverage of Cenozoic sedimentary record we can use. And one strong point of ocean drilling sample is strong, robust chronology or age model. We can decide age quite precisely. For example, for relatively young time for the past 5 million years, we can use oxygen isotope curve matching. And even deeper time, we can use plankton fossils biogeography. It's a dating based on the time of extinction and speciation of individual species we know. Then we can know the time of sediment. So this chronology is quite precise, much better than, for example, molecular phylogeny or molecular clock. So yeah, it's good for our paleo research into deep past. And yeah, here we can use microfossils. And we have different types of microfossils. Some are benthos, living on the ocean floor, like Benthic Ostracoda and foraminiferans. And we have planktons, like diatoms, calcareous nanofossils, and planktic foraminiferans. And also small parts of big animal. This is shark denticles. So we have many different kind of small fossils abundantly included in sediment core. So we can study hundreds or some thousands of specimens relatively easily. And in this presentation, I'm mainly talking about Benthic Ostracoda. It's my pet animal. And I also talk about planktic foraminiferans later. It have really beautiful fossil record. And this is one example of marine biodiversity. And this is southern ocean deep sea organisms. So we have beautiful biodiversity in species and also different beautiful morphology. But if we get into fossil record, almost nothing is preserved of entire marine ecosystem, and only limited microfossils is direct evidence of past ecosystem in the ocean. So we should use microfossils to understand broader deep time past ecosystem. It's a reason why we use microfossils to study the past. It's almost the only way, or one of the few ways. So I say we are extremely lucky. We have really good deep ocean drilling program to get the continuous long time record with precise chronology. And we have microfossils of both benthos and plankton. And so we can be sort of biologists having time machine. So this is the topic of today's talk. But this perspective is relatively underexplored from biological perspective. So people use more deep sea drilling for reconstructing climate and environment rather than ecosystems. So let's start from long-term Cenozoic through biodiversity pattern reconstructed from microfossil record in deep sea drilling program. Again, this is entire Cenozoic for the past 66 million years. And this is climatic curve. So this side is warmer climate and this side is colder climate. And this is biodiversity curve, species richness of different types of organisms. And we have really good data of planktic foraminiferan zooplankton biodiversity, and two different curves reconstructed by two different methodology, but they basically show similar or same trend. So let's see blue, light blue curve here. So after KPG extinction, strong diversity decline. After that, we have quite high biodiversity during greenhouse Eocene time. And after Eo transitional cooling, we have declining biodiversity. And it's increasing again during Miocene, and we have highest diversity in Pliocene. And this shows striking similarity with climatic change in general. Especially we have high diversity during super greenhouse Eocene time, and during abrupt cooling of Eo transition, we have rapid species diversity decline. So we see strong climatic impact on this long-term scale biodiversity change in planktic foraminiferans. And we see similar trend in other data, like not microfossil. This is a shark data based on Fabian Condamines paper, and showing striking similar trend with foraminiferal diversity curve. Even terrestrial record is neotropical paleo species richness, showing high Eocene biodiversity similar time with planktic foraminiferans, and the distinct cooling event is marked by distinct diversity decline, both in shark and paleo. And we also have another microfossil record. It's a calcareous nanofossil. Coccoris 4 record also shows similar pattern, like a Eocene peak and Eocene Oligocene diversity decline. And in contrast, they are basically calcareous microfossils, but silica microfossils like a diatom show opposite trend because they tend to like cold water, so they prefer cold environment. So they are in low diversity during warmer time, and they have higher diversity during colder time. And cetaceans show similar diversity trend, and some people say cetaceans and plankton evolved together, so they show similar diversity trajectory. And one more important thing, today is a Humboldt Day, so it's good to speak about latitudinal diversity gradient. Probably everybody know here, it's a high diversity in tropics and lower diversity in higher latitude, like Antarctica or Arctic. And it's true in deep sea as well. We have deep sea latitudinal diversity gradient. In deep sea, we have higher diversity in lower latitude and lower diversity in higher latitude. And by using deep sea drilling sample, we can look into when such trend started in geological past. And you can see here, this is deep sea foraminiferan data. And the red one is equatorial tropical data. And blue one is Southern Ocean, so Antarctic polar data. So we have now clear latitudinal diversity difference. So equatorial diversity is much higher than polar diversity right now. And the same trend continues into Quaternary, Pliocene, entire Miocene, and Oligocene. We still have higher tropical diversity and lower polar diversity. But from here, we don't see clear pattern anymore. During earlier Eocene, we have quite similar biodiversity, both in tropics and polar latitude. So this time is probably the onset of latitudinal biodiversity gradient. And I tend to think opening of Drake Passage and resulting circum-Antarctic seaway development and global scheme of present-day oceanography may cause present-day like latitudinal diversity pattern from here. Especially it's interesting, the onset of latitudinal diversity gradient is much earlier than Eocene-Oligocene transition. So something other than climatic change is probably responsible for this beginning. And apart from the global trend, let's narrow down the scale a bit into regional record of shorter time scale. And here is North Atlantic Ocean. And this is one of my favorite ocean because of beautiful calcium carbonate preservation. And also because I did a postdoc here in Washington, DC area, 5 years in USGS and Smithsonian's Natural History Museum. So at that time, I started North Atlantic deep sea paleoecological study. And I'm introducing mainly the result from these 3 sediment core locality. And we have beautiful record from here, ODP 925 in equatorial Atlantic Ocean. And this is global climatic curve on the top panel. And we have deep sea biodiversity curve of Ostracoda at the bottom. And we see beautiful glacial interglacial climatic cyclicity, like here is warmer time and colder last glacial, and warmer last interglacial, and glacial interglacial, glacial interglacial again. And this curve is strikingly similar to deep sea biodiversity curve. So all of the peak in biodiversity is well corresponded to warmer peak in climate, strongly indicate climatic impact, global climatic change impact on deep sea ecosystem. It's interesting because deep sea is far away from ocean surface, but still global climatic change have strong signal on deep sea biodiversity. And the same is true in shorter time scale. For example, it's important to think about decadal or centennial time scale because it's our time scale. When we think about like global warming and its implication, we are worrying about the time of like our children or grandchildren's generation. It's a kind of decadal centennial time scale future. So I studied high time resolution record in decadal, multi-decadal or centennial timescale for the last 20,000 years. And this is two biodiversity curve of deep sea benthic foraminiferans, and one in northeastern Atlantic and one is northwestern Atlantic. So this is south of Iceland, and this is next to Florida on South Carolina slope. And we have two distinct abrupt climatic events known as Heinrich event 1 and Younger Dryas. During this event, we have strong diversity signal here and here in both sediment core. So we have distinct double spike, Heinrich event 1 here and also here. And we also have substantial diversity decline during Younger Dryas. And these two core have almost 4,000 kilometer distance between. So it's surprisingly similar trend. And it's additional evidence of climatic impact on deep sea biodiversity, even in shorter time scale and rapid changing climate. And if we conduct some simple regression modeling and model selection with several environmental factors like temperature, productivity, seasonality, we consistently see significance with deep water circulation or temperature term. So yeah, I think temperature is the main controlling factor of deep sea biodiversity in this kind of time scale, both long glacial, interglacial cyclicity, and also decadal and centennial time scales. And we can see not only change with time, but also we can see time slices if we are lucky. And this is deep sea latitudinal diversity gradient in present day orange, and the last glacial maximum of 20,000 years ago as blue. And for comparison, I put the same thing with pelagic ocean. It's a planktic foraminiferan data in the North Atlantic Ocean. The same, orange is present day, and blue is last glacial of 20,000 years ago. So pelagic ocean is straightforward. We have high diversity plateau in tropics, both in present day orange and glacial in blue. And we have lower diversity in Arctic Ocean. But we have steeper gradient in glacial time compared to now. And it's a bit different in deep sea. We see beautiful straight line of latitudinal diversity gradient in present day. But in the last glacial time, we don't see any statistically significant relationship with latitude and species diversity. So we may have latitudinal diversity gradient only in warmer time, and during glacial time it collapsed or weakened. We have something like this again through glacial, interglacial cyclicity. And I tend to think it's probably temperature play a role, obviously for pelagic organisms, because steeper glacial latitudinal gradient is because of steeper temperature gradient with latitude, because of polar amplification. So during glacial time, higher latitude is much colder, but tropical temperature didn't change much. It's probably the reason. And even with deep sea, we have temperature difference with latitude present day. For example, Arctic temperature is colder than Atlantic Ocean temperature, but paleoclimatic studies shown last glacial maximum water temperature is quite homogeneous and no real latitudinal structure. So it may be a part of reason why we see clear latitudinal diversity gradient only in the present day, but not in the glacial maximum. And the study I've been talking about today is a rather old study. I started when I was postdoc, but after that, in January 2011, I moved to Hong Kong, and I started tenure track position there. And after that, I've been here in Hong Kong already almost 10 years. And I worked with great people, already 5 PhD students and 1 master and 3 postdoc graduated. And we enjoy Ostracoda paleontological study globally. So I like to introduce a bit. And this is our member this year. And also now undergrad student Qing Wen is helping our research, and new PhD student KK is joining us from the next month, October. So we are growing. So in the later half of my presentation today, I'm giving a few examples, actually 3 examples of a recent study from our laboratory, mainly about spatial biogeographic biodiversity distribution. We call such present day biological data as neontological data. It's a present day information about shelly taxa with high fossilization potential. It's important because biologists usually don't decide what you study based on they have shell or no. They don't care. So often good fossil taxa have really good paleontological data, but the biological information of such taxonomic group can be limited. It's a major knowledge gap. So from this perspective, our laboratory conducted modern biogeographical or spatial study of Ostracoda. It's a shelly small crustacean, a very nice calcite shells. And the first study is done by Anna. And Anna was my PhD student, and she's now postdoc in South Korea. And this study is one of her PhD chapter, one kind of main part. And it's published in our IBS Society journal, JBI. So it's good to introduce in this event of Humboldt Day, I think. And we collaborated with German Icelandic deep sea biologist Alex, Pedro, and Julunder and Saskia. And Saskia was the chief scientist for collecting deep sea samples for this study. And our laboratory member, Bryony Hisayo, helped various aspects of the project. And my long-term collaborator, Chi Ling, helped the data analysis. So we got good bunch of samples around Iceland, and southern part is North Atlantic proper, and northern part is Nordic seas. And then we conducted standard cluster analysis and NMDS, and we recognized 3 distinct assemblages. We can see here in the different color. So we have distinct North Atlantic assemblage and Nordic Sea assemblage and shallow marine assemblage in between, because we have shallow ridge between Greenland, Iceland, and North Sea. So this shallow ridge works as an effective barrier. Then Atlantic fauna cannot go north, and Nordic Sea fauna cannot go to south. We have good separation of fauna. It's interesting, very clear trend. And if we look into biodiversity trend of these samples, we have some beautiful pattern. Our latitudinal coverage is not so big, only about 10 degrees, but we still see statistically significant latitudinal diversity gradient. Diversity is higher in lower latitude of Atlantic Ocean and lower in Nordic Sea. And if we look into depth gradient of biodiversity, we see quite interesting trend. And yellow is the Nordic Sea, and purple is North Atlantic proper. So we have unimodal trend in North Atlantic proper with diversity peak at around 2,000 meters. And we have kind of linear trend in Nordic Sea. It's consistent with our deep sea biology understanding. Deep sea biologist says we have biodiversity peak in intermediate depths, usually 2,000 meters. But in colder place like Nordic Sea and Arctic Ocean, this peak can be quite shallow, like several hundred meters. So our Ostracoda data is quite consistent with our understanding of general deep sea organisms living now. And similarly, we can conduct regression modeling, model selection to see the environmental driver of deep sea biodiversity. And it's an interesting point. We have deep sea biodiversity debate. Deep sea biology people say temperature is important or productivity is important for large-scale biodiversity pattern. I tend to say temperature is important based on my paleoecological research with time series. Similar modeling always show supporting evidence for temperature, but not to productivity. But people working with modern distribution of living deep sea animals like bivalves or crustaceans consistently say productivity is important, temperature doesn't matter. So we are arguing, and there's no solution right now because living animals doesn't have fossil data, and Ostracoda or microfossil doesn't have good spatial data present day. So the logical next step is having good spatial data of microfossils in present day, because like bivalves, crustaceans, they don't have good fossil record in deep sea. So we have good bunch of samples here for modern ostracods and conducting the same regression modeling, model selection, and we found temperature significance of biodiversity control. So now it's good. We have evidence of a temperature control of deep sea biodiversity, not only in time series with paleoecological study, but also spatial study in the present day by using same taxonomic group. And while we see significance in productivity as well, so I don't say productivity is not important, but at least I argue temperature importance is underestimated. So yeah, so another study provided really good baseline information for microfossils, because microfossils tend not to have really good spatial data present day despite of their fossil preservation. So by adding this kind of basic data, we have strong evidence. How can I say? We can say fossil reconstruction of past ecosystem is not only for ostracods, but applicable for broader deep sea ecosystem or biodiversity. So it's quite important basic data by Anna. And we have similar, but not deep sea, but shallow marine study done by Ruby Chiu. She's also my former PhD student, now doing postdoc in Europe, in Czech Republic. And we collaborated with United States ostracod specialist Tom Cronin and Laura Gemelli in USGS, and Jean Hunt in the Smithsonian Institution, and Chiling helped our statistical analysis. This is the main part of Ruby's PhD thesis. And again, it's on Journal Biogeography, so it's good to introduce in this Humboldt Day event. And this is main result. And we compiled all the available data of eastern coast of United States, Greenland, and Antarctica. So basically, northwestern Atlantic Ocean on the continental shelf. We have beautiful latitudinal diversity gradient, higher diversity in the tropics, and lower diversity in the Arctic Ocean. So basically, big circle with reddish color mean higher diversity, and smaller dots with bluish color mean lower diversity. So we have much higher tropical diversity than polar diversity. And this is the same thing with different weight. And we divided diversity into different aspects, gamma diversity, beta diversity, and alpha diversity. So gamma diversity shows stronger gradient than alpha diversity. It's because alpha diversity is local diversity, so they have a lot of noise. But gamma diversity is kind of large scale regional diversity, so they don't have too much noise. So usually they show stronger pattern with latitude. So again, diversity is much higher in tropics compared to Arctic Ocean. And one interesting aspect of shallow marine latitudinal gradient is out of the tropics model. It's proposed by paleontologist David Jablonski by using bivalves data. They say species tend to originate in tropics and then tropical species or genera gradually expand their distribution into higher latitude. Is a reason why tropical diversity is much higher than extratropics diversity. We can see interesting supporting evidence by studying beta diversity in our Ostracod data. So we divided beta diversity into two different components. It's a nestedness and turnover. Turnover is a species replacement with an environmental gradient. For example, species A coming and then they disappear, then replaced by species B, C, again with some, for example, thermal gradient. And nestedness is along with environmental gradient, random disappearance without any replacement. So it often represents sort of source and sink dynamics. And this is the relative importance of nestedness along latitude. This is species level and this is genus level. So they have good latitudinal gradient. So nestedness is much lower in tropics and much higher in Arctic Ocean. It's consistent with out of tropics model because species originated in tropics, so they have not much disappearance. Of course, it's originated here, but along the gradient of latitude, that means temperature getting cooler, so they have random disappearance gradually, and then it's the highest nestedness in Arctic Ocean. It's supporting evidence of OTT model. And again, we conduct regression modeling and model selection. Temperature is a significant factor. So it's quite interesting. All time scale and all spatial scale, like entire Cenozoic and glacial interglacial scale and centennial scale, and also present day spatial pattern, all show temperature significance similarly. So temperature is probably the main driver of marine biodiversity, not only present day, but also in the geologic past. And a bit looking into more detail, because OTT model is proposed by bivalves, and we have Ostracod data, so we compare Ostracod, so small shelly animals, and bivalves, big shelly animals, regarding the details. So this is comparison between Ostracod and bivalves regarding distributional range. It's interesting. At the genus level, we have similar distributional range between bivalves and ostracods. Like in mid to higher latitude, about 5,000 kilometer distributional range, both in bivalves and ostracods, and it's narrower in lower latitude. But if we look into species level, ostracod distributional range is much narrower than bivalves. It makes sense because ostracod is smaller, and also they are direct developers. They don't have plankton dispersal stage, so they have more endemic distribution. And also we can compare species richness trend between bivalves as yellow and ostracod as purple. And we have much stronger latitudinal diversity gradient in bivalves. And we still see diversity gradient in ostracods, but it's noisier. It's also no strange pattern, but quite well consistent with, for example, classic meta-analysis done by Helmut Hilbrand in 2004 by using available data for all marine and terrestrial animals. They show stronger latitudinal gradient in larger animals and noisier gradient in smaller animals. So this ostracod bivalves comparison is quite consistent with our broader understanding of latitudinal diversity gradient. So ostracod is not strange animal, but they nicely follow general ecological rule. And one more interesting thing is here. This is per genus species richness. So that means how many species per genus. And bivalve data yellow is quite consistent with out of tropics model because it's lower in lower latitude and it's higher in higher latitude. It makes sense because according to OTT model, species originated here in tropics. So they should have fewer species per genus because it's origination place. But gradually moving out to higher latitudes, they have more species in each genus. We see partially consistent pattern in Ostracoda because past genus species richness is low in tropics compared to mid-latitude. But the richness decreased again in Arctic Ocean. So this implies we may have not only out of tropics mechanism, but also we may also have out of Arctic from north to south process. So we can explore such possibility by plotting age of genera in our Ostracoda data. So this is age of each genus of Ostracoda in million-year timescale. So some genus originated in older than 400 million years ago. It's quite old, but some genus is originated quite recently, like 100 million years ago or 10 million years ago. And this is tropical data, and this is subtropics, and this is temperate and Arctic. And basically tropics, subtropics, temperate have similar, like median age of 56 million years old over origination time of genera. But in Arctic Ocean, we have quite young genera, like median age is just 23 million years ago. And most of the genera originated here in youngest time being. So yeah, it's supporting our idea about out of Arctic because it's young, so originated in the Arctic, and then may gradually disperse to south to subtropics or temperate region. And this is the last slide of Ruby's study showing distribution of different species of ostracods. So we have quite nice tropical fauna and subtropical fauna and temperate fauna and Arctic fauna. So again, this is quite good baseline data, probably most comprehensive ostracod data present day right now. So by having this result and data, having standard latitudinal gradient present day, and they basically follow general ecological rule consistent with meta-analysis. That means ostracod result from deep time is not only for ostracod. We can apply such general conclusion for broader marine ecosystem. So really good job. And last example is my own study. With my long-term collaborator, Chi Wei in National Taiwan University, and also Mikhail Kuchera and Karsten helped with a beautiful planktic foraminiferan database. Mark Costello, Derek Tittenser, Wolfgang Kiesling, Tim Bombrake, and Buntaro Kusumoto, and Yasuhiro Kubota helped interpreting data, discussion, and bringing this project pushing forward. And Clay and Lan, they're excellent paleo-climatic modelers who helped with paleo sea surface temperature, and Andres Baselga helped with beta diversity analysis. And as I showed the example of Ruby's study and Anna's study, it's even difficult to have global scale spatial data in present day. And it's even more difficult to have such timescale, time slice, global scale time slice in geologic past. But in the case of this, we are extremely lucky. We have really good planktic foraminiferal data because planktic foraminiferans are important for paleoclimatology and paleoceanography. So many researchers accumulating data. And Mikhail did a great job compiling all data and standardizing taxonomy with beautiful quality. And eventually we have really nice global time slice, not only present day pre-industrial, but also last glacial maximum of 20,000 years ago. So we can compare global diversity pattern with glacial time and pre-industrial time. And we know well about temperature control of pelagic or planktic foraminiferan diversity. So by taking this advantage, we can do future projection of global diversity. So this is the main result. And this is global biodiversity distribution. And this is latitudinal diversity trend, equator and northern hemisphere, southern hemisphere, and diversity level of last glacial maximum 20,000 years ago, and the present day pre-industrial time. And this is future by using IPCC business as usual scenario, no restriction of greenhouse gas emission. So let's see this present day pre-industrial data. We see beautiful latitudinal diversity gradient, but with strong diversity dip in the equator, showing bimodal latitudinal diversity gradient. And recent marine biodiversity study increasingly shows latitudinal diversity gradient is bimodal with low biodiversity in equator. But we didn't have really good idea about why. One possibility is tropics is already too hot, so diversity is low. And another possibility may be higher diversification rate in deep time in tropical edge in the mid-latitude. But we basically have present day data, so it was difficult to differentiate such mechanisms. But now we have last glacial maximum and present day, so 2 time slices comparing, then we can say more about some mechanism. Let's see last glacial maximum data. We have unimodal trend. We don't have any equatorial dip in biodiversity. So that means equatorial dip or bimodality have been developed from last glacial maximum to pre-industrial time for the past 20,000 years. So that means it's ecological timescale. So from 20,000 years to present, we didn't have any extinction or speciation in planktic foraminiferan. So it should be explained by distributional shift related to thermal or temperature needs. So it's, yeah, so it's true. Tropics is already too hot for marine biodiversity. And at the same time, we can say this equatorial depression is not purely by human being, because it's pre-industrial data, and we already see latitudinal diversity gradient bimodal. So it's happened before anthropogenic greenhouse gas emission. So probably after the last glacial maximum, we have the glacial Holocene warming, and then we already started tropical depression biodiversity. But it does not mean tropical diversity depression is not related to human being, because if we do normal operation for producing CO2, we have much stronger diversity depression in the equator in near future. So obviously we need to reduce our CO2 emissions. And a few more slides. And yeah, biodiversity temperature relationship, glacial and present day. So optimum temperature is about 20 degrees Celsius. So we have linear positive correlation temperature and diversity, but after 20 degrees, it's decreasing, both in glacial time or present day. So like 25, 30 degrees Celsius is already too hot. And this is diversity change from glacial to pre-industrial and pre-industrial to the future for business as usual scenario. And this is more better scenario. We reduce CO2 emissions substantially. So surprisingly, even compared to last glacial to present day, about 20,000 years ago, tropical diversity depression will be stronger in near future between pre-industrial and 2100. So we also see a kind of diversity moving up to the mid-latitude. In last glacial maximum and pre-industrial, we see only in middle latitude, lower than 40 degrees south or north. But in future, we have much stronger such diversity shift into higher latitude beyond 40 degrees south and north. But if we reduce CO2 emission in the future, we can keep such change minimum. And we still have time.
Hi, Moriaki. We are already 9:54.
Okay.
So it would be great. Well,
sure. I'm finishing in 1 or 2 minutes.
Oh, yes, sure. Okay, fantastic. Thank you.
Sure. So we also see biodiversity, beta diversity pattern, but I may skip this one. And yeah, again, this is the same figure. So we are already in pretty hot place especially in tropics. It's already too hot, and in future we may have substantial diversity decline. And yeah, so this is actually happening. This is a conceptual figure done by William Chen and Daniel Poli. In temperate ecosystem, we have tropicalization. So like a colder water fish blue, temperate water fish orange, and warm water fish red. So in the temperate place, due to global warming, we have tropicalization. We have more warm water fish in temperate latitude. But in tropical ocean, we don't have super tropical fish because such temperature doesn't exist now. So by global warming, we have less species in tropics. Only a few warm adapted species can survive, but other species may be gone. So we are actually seeing this, and we provide direct fossil evidence from plankton foraminiferans. And one more thing, taxonomy is important. I also do taxonomy. This is my recent taxonomic monograph of deep sea ostracods. And to do good biogeography, we need not only good data analysis, but also good taxonomy. We need both. And take home message. And I have one more slide, but this is almost final. So fossil is good to describe past biodiversity. And we can reconstruct not only really deep time, but also relatively short centennial timescale stuff. And we have pretty much climatic sensitivity, and temperature control is surprisingly strong. And it's important to have both paleontological and also neontological data. And we need both good taxonomy and good data analysis. So we have a kind of biological time machine. And final slide, I like to emphasize more collaboration with different people, like a paleontologist, biologist, biogeographer. We should work more together. Actually, it's already done really nicely in IBS, our society. As you can see, this event, we have many good paleo talk. So we are already quite multidisciplinary people. And I also work with several global initiatives and networks. We need to put more such an interdisciplinarity in global network. Like I'm working with DOSI, climatic change team. I take over a new co-chair for climatic change working group in DOSI, Deep Ocean Stewardship Initiative. And I also work with Biodiscovery, Future Earth, and I'm helping IOC UNESCO, and I'm not directly working on, but pages are really excellent paleoclimatological global network. So we should have nicer collaboration and multidisciplinary effort with these guys' global network. And we are doing quite good. So I'm optimistic about our future. By doing this, we will see nicer understanding over our biosphere, not only present day, but also geological past. Sorry, it's a bit long, but thank you for your attention. And yeah, let's have quick question time.
That was fantastic. Thank you very much, Moriaki. Great message at the end, like trying to collaborate more with different disciplines. That's great. So we still have time for some questions. I think that Miguel as well put a message on the chat saying that if you wish to ask some questions, you could do it posting the question and answer, or raise your hand. So any questions? I'm not sure, Moriaki, if you see the comments on the chat, but there are like great work. Thanks for this nice talk. Really enjoyed the talk. Thank you from a lot of different attendees. So Let's wait a little bit. Is anybody willing to ask any question? Well, I have questions as well, so I will start. Oh, so we have one. So let me read it. So the first question, it is, I have a question, if there is time. Yes, right. Would you expect the same tropical depression during hot times in the past when the configurations of the continents was completely different? That is a question from Aaron or Dea.
Yeah, it's really good question, Aaron. Probably, yes. We don't have really nice quantitative data in deeper time. But for example, at least Eocene time, like 40 million years ago, I tend to think tropical diversity is lower than mid-latitude. So I think it's true, not only real recent past, like last glacial maximum or present, but also deep time. I think it's generally true.
Okay, we have another question, and I think that we have a little bit of time left. It said, we observe that latitudinal shifts are happening in modern marine taxa, with deep sea taxa being more susceptible, possible due to adaptation to narrower thermal niches. How could we, theoretically, disentangle the influence of thermal stress and shift in productivity as a driver for these shifts? That would be the first question. And then there is another one that says, also, won't this decrease the slope of the latitudinal diversity gradient as species move polewards? Would a very hot well have the same flat gradient as an ice age scale?
Yes, for the first question, basically we can compare temperature and productivity aspects a little bit separately by using time series, because temperature show more global predictable pattern, like warmer during interglacial time and colder during glacial time. But productivity signal is more regional specific. Like in some place, productivity may be higher in interglacial time, but can be lower in some other place during interglacial time. So it's not truly theoretical, but in practice, we can compare different regions with data of temperature and productivity. We conduct simple regression modeling with a multiple variable we can evaluate. Is this answering this question correctly? And for the second question,
the one about the slope of the latitudinal diversity gradient?
Yes.
As
so
forwards? That's the, yeah.
I'm not so how. Let me see. So very hot world have same flat gradient as ice age case. If we assume linear relationship between temperature and diversity, if we have like a flatter thermal gradient in super hot time, we should have flatter latitudinal gradient. But the productivity is probably not, relationship is probably not linear, but like this, too hot temperature is no good. So maybe having flat, but with some depression in the equator, it's just my casual guess. I'm not so completely sure.
Excellent. I think that then we are done with questions, and we should start wrapping up and saying thank you very much, Moriaki, for this great presentation, and everybody for attending this session. And I hope to see you as well in other sessions. Miguel, would you like to say any final words?
Well, just a final thank on behalf of the International Biogeography Society. We were really happy that you accepted to participate, and like you said, bringing a paleontological sort of perspective that is common to a lot of the work that people do in biogeography. So thank you very much for helping. You were one of the first ones to accept, so we really didn't have much to show when we first contacted you. So it was great that you accepted it. Thank you very much for doing it. And to everyone else watching us, please continue to support all of the other events. There's still several events happening today and then until Saturday. So we've been having a lot of support from people from all around the world. We haven't been really sleeping lately. So keep joining us for the events, and let's make this a real celebration. Okay. Thank you. See you guys somewhere.
Yeah.
Thank you very much. I really enjoyed. Thank you.
Thank you. Bye-bye.
Bye.
Bye.